Hello students, today we will discuss about the classification chapter of NCERT. So you all know that there are kingdom, right? So first classification was made by Aristotle. Aristotle. He normally observed the morphology, and based on the study of the morphology he divided plants into three. What you know: herb, shrub, and the tree. So herb, shrub, and tree came from him only. After Aristotle, Linnaeus, who has discovered the nomenclature, so that Linnaeus has made two kingdom classification. What are the two kingdoms? One is plantae and another kingdom is animal. Plantae and animalia. Now, after that, in 1969, there are another kind of classification came. So I have to share the entire screen first. Recording is already in progress. So now you can see the document. Okay. In 1969, the five kingdom classification came. And who proposed it? R. H. Whittaker. R. H. Whittaker. What are the five kingdoms? The five kingdoms are Monera, Protista. You will see each of them separately. Monera, Protista, Fungi, Plantae and Animalia. Plantae and Animalia. So you now know that Aristotle. What are the bases of classification in case of Aristotle? Based on morphology, I told. Now, what are the bases in case of five kingdom classification? Bases of five kingdom classification. Suppose if I tell you to divide your school classroom, how you can divide the students either based on sex or based on height. Similarly, the basis of classification of what? The basis of classification mainly based on five main characters. One is cell nature, whether it is prokaryotic or eukaryotic. After that, body organization means whether tissue is there, whether organ is there or not. Third one is reproduction. Fourth one is nutrition. And fifth one is phylogenetical relation. Phylogenetic relation. That means evolution, whether which one is simple and which one is complex. Okay. Now you have to also see the comparison between these different kingdoms. So what I am doing, I am making a table. Okay. So in that table, five will be there, and based on certain characters, we will compare. So first one is character. Based on which character we are comparing. So first kingdom is what I told you, Monera. Next one is Protista. Third one is fungi. I am writing in short form. Fourth one is plenty. And the last one will be animalia, right? So I am including another column, animalia. Okay. So first I told these five kingdom classification is based on cell structure also. So what kind of cell it is? So in case of Monera, the cell is prokaryotic. All others are eukaryotic. Then now coming to the body organization. Coming to the cell wall first. Cell wall. Okay. So you know that in animalia cell wall is absent. In case of plant, the cell wall is made up of cellulose, right? Cellulose. In case of fungi, cell wall is present, but it is made up of chitin. Okay. In case of protista, few, not all, some has a cell wall, some has cell wall, not all. And the cell wall composition is also different, completely different. Okay. So please remember that. And after that, now the coming to Monera, right? Monera, in case of Monera, what kind of cell wall is there? In Monera, non-cellulosic cell wall. Non-cellulosic means cellulose is not present in the cell wall, but cell wall is there. Non-cellulosic. So what is the made up of? If it is not made up of cellulose, what is the um, uh, chemical composition? Polysaccharide and amino acid. Polysaccharide and amino acids. Cell wall comparison is over. Now the third one I told you that body organization, right? Body organization. Okay. So in case of body organization, in case of Monera, cellular level, tissue is not at all present. Okay? Tissue is not at all present. It is cellular level. Cellular. It is also cellular. But in case of fungi, tissue cell is there, cellular is there, and some loose tissue is there. In some fungi, loose tissue is there. Cellular in case of yeast, yeast is the only fungi which is unicellular. Okay. In case of plants, tissue and organ is there. Tissue and organ is there. In case of animal, tissue is also there. Um, organ is also there, and organ system is also there. In some animal also, cellular is also present in case of, um, sponges. The cellular organization is present. Okay. So the font size is very big. Okay, not an issue. I will reduce it. So cell wall, body organization. The last one we will talk about nutrition. Okay? What kind of nutrition is present? Mode of nutrition. Mode of nutrition. So in case of animal, it is heterotrophs. Heterotrophs. In case of plants, they are autotrophs. That means photosynthetic. They are autotrophs. Only photosynthesis. In case of fungi, they are heterotrophs because they are saprophytic or parasitic. I am not writing. Okay. In case of protista, okay. In case of protista, they are actually autotrophic or heterotrophic. Okay. In case of protista, if they are autotrophic, they are photosynthetic. And heterotrophic is also there. In case of Monera, they are also autotrophic and heterotrophic. But in case of autotrophic, they are photosynthesis or chemosynthesis. Chemo, chemoautotroph. Okay, chemoautotroph versus photosynthesis. You, I think, know the difference. Okay. Um, in mode of nutrition, in another chapter, you will be able to understand the difference. So this table is over. Okay. Now what we will see, the drawbacks drawbacks of previous classification. See that. Please remember that you all know that there are different kinds of atomic model, right? There are different kinds of atomic model. First, Rutherford atomic model, then Bohr's atomic model, then modern atomic model, right? Why this kind of model came? Because the previous atomic model has a disadvantage or drawback. Similarly, why Whittaker classification came into picture? Because the previous classification must have some drawback. So we have to know that, okay, we have to know that what are the drawbacks of previous classification. What happened, that please remember that Monera, I told you, Monera is the only prokaryotes. All other protista, fungi, then plants and animals, all are eukaryotic. And please remember that Monera is prokaryotic, protista is eukaryotic but unicellular. Protista is eukaryotic but unicellular. In case of fungi, they are eukaryotic but multicellular. Okay, plants and animals, eukaryotic, multicellular. So, are you getting the point? Prokaryotic to eukaryotic, we are moving. Unicellular to multicellular, we are moving. That means prokaryotic is the simplest form, eukaryotic is the complex form. Unicellular is the simplest form, multicellular is the complex form. That means phylogenetic relation is there, evolution relationship was there. So the drawback of previous classification was like that. They don't differentiate it in that way. They haven't taken phylogenetic relationships. Okay, so they put the first point of drawback. I am not writing, I am just telling you. You just keep some notes, okay, and you write there. So what are the drawbacks? First drawback is they put prokaryotic and eukaryotic in one group. If you take what was the previous classification, two kingdom classification, right, by Linnaeus. So plant and animal, they have he has just made. But in plants, they included the fungi also, which is completely different. So these are the drawbacks, right? In previously, the Aristotle only differentiated the plants, not animal, no grouping of animal. So these are the drawbacks. Now coming to Monera, we will discuss his kingdom separately. Now coming to Monera. So please remember what I told you first. Character of Monera. Monera is prokaryotic. Okay. So prokaryotic means bacteria. Bacteria come under Monera. Whatever bacteria is there. So based on shape, we can differentiate the Monera. Differentiate the different bacteria. Okay, so I can make a table for your understanding. So if you write types of bacteria, based on shape, based on shape. Okay, so you will find four types. Okay, you will find four types. One type is cocktail. COCCI. The singular is coccus. Cocci is plural. So cocci means whatever bacteria is circular, they comes under cocci. The next one is bacilli. The next one is bacilli. So bacilli means those bacteria who are normally, actually I will type those things whose spellings are little bit complicated. Otherwise I will only talk. That is better. Okay. So bacilli is over. Next one is uh, spirilla. Okay, not spidulina. Spidulina is different. Okay, spirilla. Bacillus means box-shaped bacteria. Spirilla means those bacteria which are spiral in shape. Okay, and the last one is Vibrio. Vibrio cholerae. Right? Vibrio. Vibrio means those bacteria who are comma shaped. Okay, those bacteria who are comma shaped. Now, Monera bacteria also, this is the based on shape. Now, based on nature, we can divide the Monera, okay, based on nature. So, the next table which I am going to show you is that um, shape Monera has mainly two types. Monera has how many types? Two types. This Monera is a kingdom, right? So Monera kingdom is differentiated into two types. The first type is eubacteria. Eubacteria. And the second type is archaebacteria. Remember that archaebacteria are ancient bacteria. That means they came first. And eubacteria are true bacteria. Okay, true versus ancient. Now, archaebacteria, eubacteria doesn't have currently in your syllabus, eubacteria parts are not there. Archaebacteria is further divided into three classes. One class is thermoacidophiles. Thermoacidophiles. Next one, methanogens. Methanogens. And the last one is halophiles. Halophiles. Let me tell you first the eubacteria. Right? Whatever green algae or cyanobacteria are there, they comes under eubacteria. Okay, let me increase also in size. So cyanobacteria comes under, or blue green algae comes under eubacteria, and they are similar to plants. That means like plants, they have chlorophyll as a pigment. So cyanobacteria is over, eubacteria is over. Now coming to the archaebacteria. Okay, please remember that the difference between eubacteria and archaebacteria, the main difference is cell wall structure. Eubacteria cell wall structure is very simple, but in case of archaebacteria, the cell wall is specialized. Why specialized? Because because they live in very extreme condition. That's why their cell wall must be very rigid so that they can survive on this extreme condition. Now I will tell you that what are those extreme conditions. Can you see that? First is thermoacidophiles. Okay, so thermo means anything related to heat, right? So thermoacidophiles live in hot springs. If you go to hot springs, collect bacteria samples, you will find only thermoacidophiles bacteria, no other bacteria. In case of methanogens, you know that cow, buffalo, those kind of domestic animals, they produce the methane gas. Why? How? These methanogens are present in their gut. Okay, and ruminant um, stomach, they are present methanogens. Okay, normally marshy areas, they are present, and halophiles is present in the salty area. Okay, so that's it, nothing more. Now you have to also know some economical importance of those bacteria. Economical importance of those bacteria. So economic importance. The next learning outcome. Economic importance. So one economic importance is that bacteria are used in production of many things. Production of medicine, and in our economy it is required. The next one is that some bacteria, those cyanobacteria or blue green algae, they have a specialized cell structure called heterocyst. Heterocyst. This helps in nitrogen fixation. One some examples of cyanobacteria are Nostoc, Anabina, etc. Okay, so these produce, and you know that bacteria decomposers. So three main points you can mention. Okay, so microbes in human welfare. Okay, this is a chapter in class 12. In this chapter, you will be able to understand the different bacteria names, which bacteria is used in which industry. So now it is not required. So one point will be heterocyst or nitrogen fixation. Next point will be decomposer as a decomposer. The third point in your syllabus, there are other so many economic importance, but in your syllabus, 3 points are there. The third point will be you can write that different industries, different bacteria are used. Now, one special bacteria you have to know, that is mycoplasma. Okay, so mycoplasma is the smallest cell. If it is small cell, it doesn't have no cell wall. It can survive without oxygen, and they are pathogenic in nature. So you have to only remember these four characters of mycoplasma. Now the question is why four characters? How the questions will come? Either they can ask that define mycoplasma, 2 marks. Okay, in 2 marks, 4 points is must. Either they can ask a question, write two characters of microplasma. In that case, two character is enough. You don't need to remember the four characters. But in case of MCQ, there is the trick. They will ask which of the following is not a character of mycoplasma, which one is a character of mycoplasma. So if you don't remember all of the characters, you will not be able to solve those kind of tricky MCQ questions. So mycoplasma, four characters are mentioned in your NCERT. You have to remember all the four characters. Okay. So now the monera is over. Now coming to protist. Okay. Now coming to protist. Protista, what I told you, they are eukaryotic but unicellular. From fungi, eukaryotic multicellular starts. Okay. So again, make a table for protista. So make a table for protista. How many groups is under your syllabus? So protista is divided into mainly five. Okay. So let's make it Protista, the kingdom name is Protista, and it has four, five groups. First group is chrysophytes. Chrysophytes. Next one is dinoflagellates. Dinoflagellates. Third one is dinoflagellates is over. Third one is pro. That means protozoa. They will come here. Okay. So protozoa. Third one is fourth one protozoa. The fourth one will be uh, protozoa is what euglenoids. Euglenoids. And the last one is slime. Emo-LDS, not emo-ULDS. Okay? Sorry. Here's emo-ULDS. Slime. Now you have to remember the characters of each one. Okay? So first start with what? Let's start with chrysophytes. Okay. Chrysophytes, five character. Please remember. First one you have to mention is that diatoms. Diatoms and golden egg. Diatoms and golden egg. Whose name, another name is golden algae, another name is desmids. Desmids. Okay, so they comes under chrysophytes. Okay, next one is their cell wall is made up of silica. Okay, and the third point is these cell walls are normally two parts are there. Each parts are fitted with each other like a soap box. Okay? They are either fresh or marine, and they are mainly fresh or marine. They make the diatomaceous diatomaceous. Okay, they are fresh or marine, and they are the last character. What you have to remember that they are photosynthetic. Okay, these points in case of chrysophytes. Now coming to dinoflagellates. Okay, now coming to dinoflagellates. Let's write in short form. So in case of dinoflagellate, the name says it all. Di means two. They will have two flagella, dinoflagellate. So one is transverse and one is longitudinal. Longitudinal. There are two flagella. Okay. Next one is they have a cellulose. Okay, in the cell wall. Okay, they have cell wall, and they make eight tiles in ocean. They are completely marine, not in fresh water. Okay, they are photosynthesis, and there can we give an example of dinoflagellate who creates those kind of red tides. So the example is Goniolax. Gun means it is gun. So gunny, gunny olax, gunny olax. LAX or LU? LAX, okay? So dinoflagellate is over. Now come to the next one, that is euglenoids, okay? That is euglenoids. How many characters you have to remember in case of euglenoids? Euglenoids also have two flagella like dinoflagellates. Okay, one is short, one is long. Okay, and instead of cell wall, they have pellicle. Okay, instead of cell wall, they have a pellicle. Okay, and pellicle is normally made up of protein. Okay, one short, one long, it already told. And in case of euglenoids, okay, in case of euglenoids, they are normally, if they are phototrophs, if they are phototrophs, they have similar pigment like plants. But they also, if the light is not present, they act as a chemoautotroph. So autotroph has two: photoautotroph, chemoautotroph. If light is there, it is photo. If if light is not there, they will produce their own food from chemicals or breaking down of chemicals. That's why they are called chemoautotrophs. Okay, and nucleonoids normally they are found in uh, not in marine, they are found in fresh water. Okay, so these many points are there in case of nucleonoids. Okay, so fresh water and stagnant water, fresh plus stagnant, please. Why I am giving it much more important phrase stagnant? Because normally what happens, which one of the following is not a character of Illinois? They will show, they will give this kind of very complicated options like they are present in fresh water, they are present only in marine water, they are present fresh plus marine water, they are present fresh and stagnant water. Okay, stagnant means what? Suppose in your house, a stagnant water is there. No rain happened. The stagnant water is present for nearly 1 month. So in that case, you will be able to find this kind of lymph nodes. Okay. In ocean, you can't find fresh water. That means river. Okay, euglenoids are there. But in marine, dinoflagellates are there. Okay, these tiny characteristic difference you have to know. Why? Because freshwater euglenoids can't survive in ocean. Okay, it is very critical, though it seems that fresh, stagnant, what is the importance? But no. Okay, so suppose there is, I told you some are parasites. So how to very quickly diagnostic a disease. So suppose there is a zoologist, you will be able to know that this guy who is suffering from fever or a disease, he lives in near the coastal area. Near the coastal area, that means euglenoids can't be there. Only dinoflagellates there is a possibility. So they will see the database of only dinoflagellates. That's how. Please remember that this is also important. Don't skip it. Sometimes it is very hard to remember which is fresh and very aquatic, stagnant water, but you have to remember it. Okay. So now coming to slime molds. Okay. Now coming to slime molds. Slime molds are saprophytic. Slime, it starts with S. No? So first point will come saprophytic. The next point is in favorable condition. In favorable condition, what happens? They make some pseudopodia. Pseudopodia. Okay, in favorable condition. In favorable condition, they produce plasmodium. Means what is the meaning of it? In favorable condition, they form aggregate. Many cell comes in contact with each other and they form a aggregate called plasmid. Aggregate called plasmid. Okay. In unfavorable condition, what happens? In unfavorable condition, what happens? In unfavorable condition, they forms a fruiting body. They forms a fruiting body via spores. And these spores has high resistance rainwater. Okay. And the only means to carry these spores is air. Are you getting the point? Suppose you are happy, you because you have a favorable condition. What we will do? You will mix up with so many friends, right? So you form a aggregate. In that way, please remember, okay? That aggregate is called plasmodium. Okay? Please remember the term plasmodium. But in case of unfavorable condition, what it does, mm, it does some bodies which contain spores. At the tip of those bodies, spores are present. And these spores, unfavorable condition, right? So it must be protected. So the spore cell wall is very rigid and they are not carried by water. So marine fresh water, most of the protista, marine water or stagnant water. But in case of slime mold, these foods are not carried out by water. They are carried out by air only. Okay, now coming to the protozoa. Protozoa are either parasitic or a predator. The parasite, you know what is the meaning of predator? Predator means produce or engulf another bacteria like that. Okay, now coming to the different protozoan variation. So protozoa has mainly four types. One is amoeboid protozoa, amoeboid protozoa, then flagellated protozoa, then ciliated protozoa, and sporozoan protozoa. Okay? So I am not making any table. Just you in your copy, please write down those things. Okay, I am just writing those important terms. So, what is amoeboid protozoa? So, based on their locomotory organ, how they move, based on them only, the protozoas are being differentiated. Okay. So amoeboid, in case of amoeboid, some examples I am giving. Amoeba is the best example. Then entamoeba, entamoeba histolytica, they comes under amoeboid. And their locomotory organ is, you know that what is the locomotory organ? Pseudopodia. Pseudopodia. Okay, yeah, locomotory organ as well as by pseudopodia they eat also. They engulf food particles by pseudopodia also. And what are the in their case also sometimes cell wall if they are present in marine, their cell wall is made up of silicon. Okay. So I told you if they are present in marine, so normally they are in fresh water, then moist soil also they are present. So amoeba, you please note it down whatever I told you. Now coming to flagellated. Coming to flagellated, okay. Flagellated, you are able to understand that the flagella is the locomotory organ, but the main point are the examples. Okay. So entamoeba belongs to amoeba, flagellated, ciliated, or sporon. This kind of question will come. So whatever example is mentioned in your book, you have to remember all of the examples. If 5 mark question comes, any one example is enough. But in case of tricky MCQs, you have to know all the examples. Okay. So the first example of flagellated is Leishman. Then next one is here. And the third one is trypanosome. Okay. I made a mistake. See that I am also making mistake. Okay. So it is quite hard to remember, but I am not going to pass the exam. You have to become a doctor. That means your memory should be much more better than me. Okay, please write down these important examples. Coming to ciliated. Ciliated means cilia is the main locomotory organ. But in case of the ciliated, there are other terms also. So there can be a question differentiate between amoeboid and ciliated protozoa. So in what are examples you can give? locomotory organ in case of amoeboid pseudopodia, in case of ciliated cilia. In case of amoeboid, the engulfment of food particle is done by pseudopodia, but in case of ciliated, it is being formed by gallate and ingestion and ingestion. Ingestion and ingestion. Ingestion means taking the food. Egestion means taking out of the food. Okay. So egestion happens with us by anus, right? In case of them, the egestion happens by cytopage. And in case of ingestion, it happens by stomach. Cystostomy. Okay. Now coming to examples, right? Examples are paramecium. Okay? Sporozoa, they don't have any locomotory organ. That means locomotory organ is absent. In case of that, what is the plasmodium, the malaria parasite. Okay, that example plasmodium, right? Plasmodium and monocystis. Plasmodium and monocystis. Protozoa is over. Now coming to fungi. Fungi character. Fungi. Already told that their cell wall is made up of chitin and they have hypha network, thread-like structure, filament-like structure. They are called hyphae. And the network of hyphae is called mycelium. Okay. What it any other character? No. Now coming to the different classification of fungi. Okay, fungi. So I have to make a table also. Okay. So first character will come. First group of fungi is called Phycomycetes. Second group is called Ascomycetes. For sac. For sac. Third one, Deciduous. They are called bracket fungi. They are called bracket. And the last one is deuteromycetes. They are called imperfect fungi. Imperfect fungi. Okay. So this is the main thing. So first character we have to go is that the nature of hyphae. Nature of hyphae. nature of hyphae in this case is accepted and sinusoidal. What is the meaning? Accepted and sinusoidal. The meaning is that suppose there is a um, building. The building has mainly four rooms. Each room contain one people. Okay, so total four people. There is wall between the rooms. That is why the four people are separated from each other. Okay. So in that case, in case of ascomycetes, basidiomycetes, deuteromycetes, the hyphal filament is septate. Walls are there. Yeah, I am not writing in case of Basidio and Deutero, same thing, septate. But in Phycomycetes, accepted is there. That means in between those rooms, the wall is not there. So the four people can come together. That happens in case of phycomycetes. In case of phycomycetes, the hyphae doesn't have any wall between them. So what will happen? Each hyphae nucleus, they will mix up together. So that means in one single hyphae cell, so many nucleus will be there. That is called sinusoidal. Normally, what you know, in a normal cell, only one nucleus is present, right? But if more than one nucleus is present, that is called sinusoidal condition. That is present in case of phycomycetes. Okay, done. Now the coming to reproduction. Their reproduction is also very different. Well, reproduction. In case of deuteromycetes, let's start with deuteromycetes. There is no sexual reproduction, only asexual reproduction, and that is done by conidia. That is done by a special spore called conidia. In case of basidiomycetes, there is no spore formation, but what happened? Two different strains. Let's write down different different strains come together for fusion. Comes together for fusion called plasmogamy. Called plasmogamy. So what is happening? Two hyphae is coming and they are fusing. There is no spore formation. Okay, and that process is called plasmogamy. Okay, because two cytoplasm is being fused, that is why it is called plasmogamy. Now coming to phycomycetes and ascomycetes, very carefully see. In case of phycomycetes, asexual reproduction happens. That is no sexual reproduction. So, but there are two kinds of spore: juice spore, which is motile, which can move, and aplanus, which is non-motile, which can't move. And they all are produced exogenous. They all are produced endogenous. So they are all produced endogenously in sporangia. Okay. But in case of ascomycetes, okay, in case of ascomycetes, A spore is formed not exogenously and endogenously. Please remember, in phycomycetes there is only endogenous spore formation. But in case of ascomycetes, endogenously also, exogenously also spores are being formed. So endogenously, the spores which are formed, these are called ascospores. And exogenously, those spores are called conidia. Same. Conidia which you have seen in case of deuteromycetes. Okay. So these exogenously, these comes under similarly asexual, but that ascospore comes under sexual reproduction. Okay, what is the meaning of this? So there are, please remember, this is very um, complicated to remember. Mm, there will be a question that in which kind of fungus group, both endogenous and exogenously formed spores can be found. That will be the ascomycetes. Again, I am revising. Next question can come endogenously ascospores are formed endogenously, exogenously, both of them, none of them like that. So please remember exogenously produced spores are called conidia. They comes under asexual reproduction and uh, those bodies which contain the conidia at the tip of them that is called conidiophore. Conidiophore. Okay, conidiophores contain conidia at the tip of them. That conidia is produced exogenously because they are produced in the tip. Okay, and that conidia they just detach from the the ascomycetes and they germinate into another fungi. That's how the asexual reproduction happens exogenously. But in case of endogenous means inside the body. Ascospores are being produced. They are not on the tip. Inside a sac-like structure, ascospores are being produced. That is why it is endogenously produced. And this comes under sexual reproduction. How? Two sacs two sacs are there. Two sacs are there. They contain ascospores. They come in contact. Two ascospores fuse and they take part in sexual reproduction. You know that sexual reproduction is nothing but fusion of two individuals. But in case of asexual, only one kind of plant is required, right? Now, Now the very important thing, introduction is over. Now you have to remember the examples. There are huge examples. There are huge number of examples you have to remember. First, phycomycetes. Let's start with mucor. Mucor is found in, if you see in the roadside some cow, cat or dog dung is there, and if one fungi is going, that is definitely me. Okay, interesting, right? In first stage, remembering that can be very exhausting, but it is interesting also, right? What kind of fungi is present in your dog poop? Mucor will grow, no other fungi. Okay, mucor is the one. Now coming to the next example. The next example is rhizopas. Rhizopas is present in case of bread mold. You keep a bread for many days open, not eating, rhizopas will grow. Mucor you will gonna be able to find. Now the third example of phycomycetes I am again repeating because they will give an example of fungi. You have to tell that which group they belongs to. So each and every example you have to remember. The third example is albugo. 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 It normally give a disease in case of mustard. Okay, three example is there in your syllabus. Now coming to 1.7 billion species are there, right? You need to remember that. Just three examples, common example. This is good, right? If somebody tells you what is there, what is growing in the mold, you can be able to tell that this is rhizopas. Don't eat it, okay? Because this fungi will contain spores, very harmful spores. Which fungi is present in our dandruff? Please find out. Okay, which fungi make the what you called our shoes bad smell from shoes, right? Which fungi, just find out. Okay, now coming to, so this is very important, daily life it is required, and this is daily life uh, examples is very useful. Now coming to, after phycomycetes, ascomycetes. Ascomycetes. What example? Neurospora. Neurospora. I haven't written those things. Just here you keep added in your notebook. Mucor, I told you the dung. Okay. Now coming to neurospora. Neurospora is used in different research purpose. Okay. Now last one, LSD drug. Have you know? Do you know that LSD drug very strongest hallucination creation drug. Okay, this is mainly produced from claviceps. 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 Claviceps purpu- purpurea. Claviceps purpurea. Okay, from here the LSD drug is being produced. Okay, coming to basidiomycetes. Basidiomycetes has so many examples. First is agaricus. First is agaricus. Whatever mushroom you are eating, that is agaricus. Okay. Next one, ustilago. Ustilago creates smart disease influence. Okay. Now coming to Puccinia. Puccinia creates rust disease in plants. All are very harmful in case of plants. Okay. Puccinia. Now there are a fungi which create toxic chemicals. The name of it is amanita. Okay. Another name is one fungi, bracket fungi. I told you basidiomycetes means bracket fungi. What is that bracket fungi called? Fusarium. Fusarium. Okay. Fusarium, it is normally found in wood. And the last example, okay, this is the last one. Please stay tuned and stay relieved because this is the last example. The last example is from their ink. You are writing, you are now noting down whatever I am telling, right, in your notebook. So this ink production is happen from a fungi. Okay, so that fungi is called cap fungi and cap fungi. And the name is, the scientific name is Coprinus. Okay, done. Now the last one, deuteromycetes. Okay, deuteromycetes examples. Okay, they start deuteromycetes. Deuteromycetes, I will start with one example, that is um, red rot of sugarcane. Red rot of sugarcane. Okay, red rot of sugarcane. Sugarcane juice you all know. So you will see that in some sugarcane there are some red rot happened, red patches in sugarcane. So that is produced by a which kind of fungi? Deuteromycetes. Okay, and that fungi, please remember that name. The name is okay. The next two examples, it is red rot, it creates red rot. The two other examples of alternaria, alternaria, almost over. Please stay tuned. Alternaria and trichoderma. They actually kill nematodes. Okay, they kill nematodes. Okay, so red rot is done by Polytotricum falcatum and Alternaria and Trichoderma, they kill the nematodes. Now, the last point is economic importance of fungi. You all know yeast produce bread. Then penicillium is also a fungi. From there, malaria medicine is being produced. Then another fungi is mm, these two um, point. Please note down. Please note down. Plant and animalia, those kingdom has separate chapter. We will be able to. The next chapter is plant kingdom. So all are over, but you know that there are five kingdom. There are three other groups also which is not included in this five kingdom. Okay? Those are virus, thyroid, lichen, and prion. Okay. These four doesn't come, you can't fit. Their characters are so unique that you can't fit those organisms into any of the kingdoms. So they are separate from kingdom classification. And even today, no scientist have ever came across to any other classification system where they include those characters. Okay, so just 1 minute. Spirions. There is a mistake actually. It is prion. Now coming to start with the virus. Okay, you know there are some viruses which kill bacteria. So those are called bacteriophages. So bacteriophage belongs to bacteria or virus. The name says it all bacteria, so it must be bacteria. But no, bacteriophage is a virus which kills bacteria. So these bacteriophages have different parts. One is head. You have to remember the diagram which is present in NCERT book. That is head. Then they have also collar. They don't have neck, they call it collar. Okay, the third one is the third one is they also have a seed. The body is called seed, and the last one they have also legs, all tail fin. Okay, they have either DNA or RNA. Okay, and they have capsid, capsid, which is a protein coat, and each unit of capsid is called capsule. They are obligate. They are obligate parasite. What is the meaning of obligate parasite? Because if you take a virus in a table, currently in a table, it is acting as a non-living creature. But once you enter the virus in any living body, it will act as a living creature. That means they are obligate, they are obliged to depend on ourselves. So that's why they are called obligate. Obligate and facultative. There are two parts of parasites. So facultative, they can stay out of the body also. That means it is optional. But in case of obligate, they are obliged. They are obliged to stay in a body. I can tell lots of things on virus, interesting things about virus, but in your syllabus, those things are not there. But obligate, whatever I am writing, please note it down. Um, Obligate parasite, then they are smaller than bacteria. These characters are mentioned in your then nucleoprotein. They have nucleoprotein. Nucleoprotein means the DNA RNA plus the protein code. They together make the nucleoprotein. And in this case, you have to remember some history also and some dates also that comes. Okay, so first date starts with 1892. Next is 1898, and the last date is 1935. 1935. So in 1892, there was a scientist who gave the name virus. Okay, what is the name? Dmitry Ivanovsky. Dmitry Ivanovsky. He gave the name. Next one, there is another guy that is M. W, and it is W. Just reverse. W.M. Stanley. He crystallized this crystallization. He crystallized a virus particle in 1930. And what is the name of this guy in W? Will. What he extracted the virus from a plant, okay, and he gave that extract to a healthy plant. The healthy plant got infected. Okay, that is how he proved. Okay, what virus he has taken? I think he has taken the mosaic virus. Okay, tobacco mosaic virus, and in his own term, he has told that virus is an infectious fluid because it is, have taken fluid from an infected plant and it made a healthy plant infected again. So, so there is a term he has given. Just remember, just find out from which country this MW Beijing case. So you will be able to understand that what is the meaning of that contagium vivum fluidum. Okay, that means the fluid is contagious. Okay, that's how we will actually try to tell. Virus is over. Now coming to thyroid. In case of thyroid also, there is a date you have to remember, that is 1971. 1971. EO. Diner, we discovered it. Please remember that the size of thyroid is similar to the virus. But what is the difference? It has no molecular weight of RNA. Okay, that means it can't have DNA, it only have RNA. And it is of low molecular weight. Okay, that is no protein code, no protein code. Same size with the virus. No, prion, it is similar. in size with the virus, but thyroid is smaller than virus. Okay, please remember that. And one example of thyroid you have to remember that in potato there is a spindle disease, potato spindle disease. Okay. Now coming to lichen, let's finish the lichen. Lichen is a good indicator of pollution, first of all. Next one, lichen is not a single entity. It contains algae and fungi. Algae and fungi combination. Okay, so what does algae? You know, algae makes their own food. So algae makes the food, it gives to the fungi, and fungi in return gives algae the shelter. So algae component is called phycobiont, and fungal component is called mycobiont. Do you know that the study of fungi is called mycology? That's why microbiome. Okay, so there is nothing else to remember in case of lichen. The last one is prion. Prion, I already told one example, one characters that is similar in size with virus. Okay, it is an infectious protein. Please remember the viroid. Thyroid is an infectious low molecular weight RNA, but prion is infectious protein compound. There is no RNA or DNA. Okay? And last one is what are the two examples of prion? This will conclude our this chapter. So the first example in case of human is chromium, Jacob, Jacob disease. Okay, this is created by prion, and in case of a cow, there is a disease. Okay, that disease is called bovine encephalopathy, bovine sponge, spongiform, spongiform encephalopathy. Okay, so these conclude our whole chapter.